Good morning, everybody. Hey, thank you, Min Lin. I will carry forth and elaborate in a little more detail on and add some granularity to some of the points that Alan made. It, his presentation was excellent, and it's going to be a hard act to follow. So, as you indicated, I have been with the FDA. I must tell you about a deficiency. I have an MD, but I am not a PhD. So I don't know how that got in there, but I, I did not lie to Congress. So I started out as a practicing doctor. I was an intern and resident in the big New York City hospitals that you've probably seen in some of the horror movies, but still had a yen for research. Wound up traveling down to Washington and was at the NIH. Did a stint of private practice where I realized I really liked that stuff. I was doing clinical trials outside of looking at patients, and then had the opportunity to join the FDA. And over the years, it's exactly what Alan was telling you about the glory of the FDA, that not only a regulatory body of interpreting various laws, but also a partnership, which led to many discoveries and the help of small startups getting going. And as you heard, those small startups and universities with some additional financial support have provided some excellent products that have met needs throughout the globe. And I had the honor of organizing some research with universities, with other sections of the government. Have to emphasize that partnership, and just as we're talking here today about partnership, and learning from each other is so important. While at the FDA, I worked with many people from Taiwan. My daughter-in-law is from Taiwan, and the granddaughter spends too much time there. So I know a lot about Taiwan, and between Min Lin and Alan, you guys gave me the traction to get even more involved. As you heard in Alan's discussion, the FDA and the U.S. government health agencies are under new leadership. There are new priorities, new regulations, a great deal of internal discussion about them now, and whether they are political or whether they're political basis, not based in science. But certainly, I'm going to talk to you about all those strong scientific underpinnings of the regulatory requirements. Admittedly, some of them will be changing. I can only tell you about my experiences. We're going to go through some of the official policy postings and maybe a hint or two of some gossip from my colleagues. Let's see. Can we go to the next slide, please? And okay, that's another hello. And hello, Taiwan. Let's go to slide 3, please. So we have a real big curriculum here. I don't think we'll be able to cover all these topics, but that's why you'll be able to contact Alan and myself for further follow-up. Certainly, there'll be a little bit of repetition here to drive home some very important points. And we all learned in school that repetition really gets us thinking. Very important to keep in mind is that cell and gene therapy products are opening new markets. The public is receptive to these new products. The regulatory approval channels are changing, perhaps becoming streamlined, and but still maintaining, hopefully, good scientific integrity. So let's move on to slide 4. The next slide, please. Yeah, so some cases in point. This is from the Wall Street Journal, as you know, a very prominent business newspaper throughout the United States, if not throughout the world. And they noted the continued discovery of new enzymatic defects that were resulting in very severe congenital illnesses, previously could not be treated, and unfortunately patients led limited, debilitated lives. But with the advances in cell and gene therapy, these conditions are now treatable, and just moving my, give me a second here. These conditions are now becoming treatable, and there's a potential for more effective treatments coming down the road. As we've been alluding to, in many cases, there's only a very small population of affected individuals. So from classic financial investment, products may not be viable, and that's why we're going to be talking about the orphan drug designation. I think reading the Wall Street Journal article would be very interesting, particularly for the business folks in the audience. They get into the various business models and what leads to profit and what leads to difficulties in pursuing this type of research. But stop the presses. Even more important to show you that the public is deeply involved. Last few days here in the United States, and perhaps folks over in Taiwan or elsewhere can get U.S. television programs, a very well-known program, 60 Minutes, that's been around for a long while, a good investigative reporting, took up the topic of cell and gene therapy and demonstrated at all levels of government and that new methodology was being developed to treat Lyme disease on the island of Nantucket. That's a very interesting island off the east coast of the United States in the Atlantic Ocean. To show you that more than humans are involved, mice were given, were instilled with a new gene that made them immune to Lyme disease, and that broke the chain cycle of the various species, including humans, that could contract this disease, which, as you know, can vary between a rash and heart and other major difficulties. So this is really important development, and they closed their report by saying in a previous century, people might have tried to kill off all the mice or all the deer, and that would just make the environment worse. Now we have a much more targeted way to deal with these difficulties. Let's move on to the next slide. You heard about this before from Alan, but just to emphasize it again, to really Stockholm your understanding of regulatory language, because as you interact with the FDA, there are various levels of guidances which are based on laws which are part of the Code of Federal Regulations. So as you become good sponsors, you will learn this important legal makeup and be able to converse very well. So if you look, you will constantly hear in your discussions with the colleagues who are still there as you develop products about Section 351, and those are products, cell gene therapy products that undergo extensive review, leading to what you heard from Alan, usually a BLA, a biological license. And the other aspect is the 361 products. Those are products that are taken from a donor and reintroduced. An excellent example of that is liposuction. People have a little extra fat removed, maybe undergoing some additional treatments, and those altered cells are reintroduced to the same patient. Amniotic fluid to treat some of the to introduce genes for some of the difficulties I mentioned earlier. That can be of a variable classification depending on how it's processed. Let's move on now to the next slide. You saw this with Alan, but keep in mind all these various techniques because they are very a la mode, and particularly the use of viral vectors, which we'll talk about in much more detail, and the CAR-T gene treatments. Again, keep in mind because I will show you some example products. Now, as we look at the next slide, number 7, if we could move to that, terribly busy slide, but this is important to keep in mind. As you heard throughout these discussions, viruses are used as vectors to introduce genes into various cells, either in or out of the body. It's fairly obvious. Viruses have been going into humans for thousands of years. They have a lot of experience on how to do it. It's important to note, you obviously can't read all that on the slide, that viral vectors vary in their own internal immunogenicity and their ability to cause diseases on their own. Therefore, as you consider a vector, you have to look at all these qualities before trying to link a gene to the particular virus. As mentioned, CAR-T, which is the chimeric antigen receptor process, we're going to talk about in a little more detail. It's gained notoriety because a couple of products already are approved and working very well. So keep those various methodologies in your mind. You as developers will certainly be working with all of them. Let's move on to the next slide, if we can. So this will give you a measure of the activity of cell gene therapy development within the FDA. It relates to the number of applications for the regenerative medicine advanced therapy designation. That's a designation I'll explain in more detail as we move along. But the important thing to note as you go down the years here, and you get to the last one for so far in 2025, you'll see there are 71 requests, 41 were granted. And since the rows don't add up down there are a lot of them pending. And this is reflective of some of the changes that are going on in the FDA, loss of some of the senior personnel. Important in that regard that between Alan and very good consultants and the development of personnel in other agencies, there will be other channels for you to get a good input as you continue with product development. So the next slide, number 9 for me. Let's talk about moving to market. Let's with the background you have that I've shown you, let's get into the real granularity of moving ahead with a potential product. So how do we move through the regulatory process? Well, let's start, as Alan alluded to, and very clearly, it's the non-clinical development. That's the CMC, chemistry, manufacturing, and controls, animal testing that you heard about. There are multiple guidances out there that will tell you about the number of tests that are required, what the FDA is looking for. All of these guidances help bring you towards the development of the first step of a new product called the IND, the Investigational New Drug Application. In the United States, once you have an IND, human testing can take place. So keep in mind some of the very important issues that the agency is looking for. If possible, a mechanism of action. We'll talk about that in a little greater detail in some really very acute developments that are occurring. You should be doing initial testing of toxicology in a relevant animal model. We'll talk about how you develop that type of model. You go, and as I was alluding to by showing you all those vectors and gene effects earlier on, you have to consider the off-target effect of the genes and the, as mentioned, the immune response of the virus, and for that matter, whether the virus somehow will become active in a particularly ill individual. Concomitant with that is a concern about long-term gene expression beyond the immediate effect that you're trying to accomplish through your R&D. Now, the FDA and bolstered by the statements that you heard from Alan, really looks for the importance of the initial dose. What type of dose do you think should be administered to humans? How can you justify that? And for that matter, how is it going to be administered? Initial dosing may be based on a scaling effect from animal studies. Some folks in toxicology can help you with those type of calculations. And again, the guidances will also help. If there's no information about an initial dose, it will be very hard to move forward with an IND. Let's move on to the next slide. So what are your interactions with the agency? How do you move forward as you're developing a product? Cell gene therapy is kind of unique and probably requires a greater interaction with the agency. Hopefully, if staff is still available, more and more meetings. You will note that there are meetings available to you to offer you initial guidance. The most early meeting is called the Interact meeting. You can see how it was put together with the various terms for the first meeting, and it's based on the on pharmacologic data. So all your expected to be able to present is the pharmacology of a product and gain some more guidance to help out with that previous slide that I just mentioned. The next type of meeting that's important is the pre-IND meeting. In that type of meeting, you are getting together all the information that was on the previous slide, and you're also developing study concepts. Most of these products are new, so you're going to be doing the first in human testing, and it's going to be single dose regimens. So in what we call phase one, as you know, the INDs are usually divided into phase 1 through phase 3, and so let's talk about phase 1, the most basic. In phase 1, you're considering a dose escalation arm. That means you try to find an effective dose, starting first, scaling from animal studies, and seeing if perhaps a higher dose will have a better effect or just the same effect as the previous one. And also you are looking at safety and tolerability, and this is performed in human volunteers. Efficacy comes later, and those are in the phase 2 and phase 3 trials. Those type of trials are very product specific, and I think beyond further discussion here, other than there should be hypothesis testing, and the goal is to move your product from its initial phases to preparation for what's called a new drug application, the famous NDA. I should also say that, again, very a la mode, after going to press and putting out the PowerPoint, there was just an announcement in the Federal Register about a new guidance on orphan drug approvals. The agency is considering a new pathway. The draft guidance is available for review. So some of the things I've said inadvertently between myself and Alan may just be obsolete after we all get to study this new guidance. Well, let's continue now along with the developmental process, and you're working on slide 11, which is an important way to think about your product. And that is the target product profile. Here, you take an investigational new drug and you outline all of the information you have about it in the various rectangles, the most important of which will be your indication statement, because that's what differentiates your product from those that are already on the market or fills an unmet medical need. As you can see, I've given you an example for treatment of muscular dystrophy, and the various options available with the new product, whether it is as good as existing products or continues to show some improvements. As you work through these boxes, you would point out how the clinical testing confirms the increased efficacy of your proposed product. Much talked about on the next slide is orphan drug designation. There's many of the cell gene therapy products would be available for this designation because they're treating gene defects or absences in a small population of people with unfortunate genetic defects. In the United States, that's defined as less than 200,000 patients, as Alan might have mentioned, because they development of these products may not be financially viable. There are various tax credits, fee exemptions, and the 7 years exclusivity where no other product can gain that same indication. But as I mentioned, there appears to be a new guidance out which may change all the information I just provided you. So the background thoughts are there. I hope this slide does not become obsolete. If we move on to the next slide, let's talk about safety considerations. Alan alluded to this, and this will play heavily into the presentation of your product, the presentation of the NDA document. For CGT products, there are some special considerations because you're looking, and it's very important to look at the safety events and trends of other similar type products. This will give you some information about the viral vectors, off-target occurrences, the shedding of the viruses, and the possible integration of the total virus into cells and a oncogenic risk. A good example for this is the treatment of hemophilia, which again came under the Orphan Drug designation. As I'll show you a little while later, there was a gene insertion into liver cells. Remember, hemophilia is a unfortunate coagulation defect where our patients, if not treated, on simple cuts, will unfortunately exsanguinate. The liver makes some of the lacking. The liver would be making some of the coagulation factors in a normal individual, and placement of genes into these liver cells has resulted in the production of the lacking coagulation factors and helped mitigate the disease. But then these patients have to be further watched for potential liver damage. And one of the, just to keep in mind, that one of the best vectors, viral vectors, seems to be the adenovirus, because it has become very popular because in and of itself it has very low immunogenicity, so it will not generate a lot of reactions. As you know, adenoviral infections have been a bane but treatable and getting by in humans for the millennia. So let's take a look, slide 14, the next one. Well, there's your goal, getting a new product out that fulfills an unmet medical need, getting it approved. So on slide, the next slide, slide 15, well, we've moved through the developmental process, and you have a new drug application, and you're going to be approaching the agency, and you have to talk about how can I get this product approved in a shorter time period, because there's information available that really indicates that this is a terrific product and will really be a game changer. So as noted along that slide are the various options available to you. We've said a couple of times that the cell gene therapies often double qualify. They can be considered for an expedited approval program. So that can shorten the time from 10 months to maybe 6 months. The agency, when they consider these various expedited programs, will often ask for confirmatory tests or confirmatory trials following the initial approval. I also talked about the regenerative medicine pathway a little bit earlier in the presentation. In addition to this being a shorter time, it provides more direct input from the agency to help developers along. Also, very, very important is a proposal by Dr. Markey, who is the current commissioner. This has not become law or in place yet, but it is the plausible mechanism pathway. This would be a conditional approval allowing the drug to be marketed without clinical trials. And then clinical trials could be done post-approval. Very controversial area, and the trade press has a lot of chatter about this. You can find a bunch of articles, and it's not clear whether this will be approved and when it will be put into effect. But certainly the major pharmaceutical companies feel this would be a game changer because they could get products out earlier and perhaps avoid the 5 or 6 years of clinical trials and $5 or $6 at least $1 million. So there you have a quick overview, 30,000 feet or 30,000 or 10,000 meter view of getting these new products out to the market. Let's just look at 1 or 2 important examples. So next slide 16. This is sickle cell disease. This has had orphan drug designation here in the United States. Sickle cell disease is predominantly in African American individuals. It's due to an abnormal hemoglobin that's in the red blood cells, which form a sickle shape, as you can see, and because they don't flow evenly through blood vessels, they lead to obstructions and severe pain and shutting down of various important organs. The 2 products that are out there are indicative of what we talked about in the beginning of the presentation. Cgt uses gene editing, and Lyfgenia uses a viral vector. With Cgt, cells have to be taken out of the body, treated, and then reintroduced. Lyfgenia, because the viral vector, is a more direct approach. If we go to the next slide, next slide, please. This is an example related to multiple myeloma, and also tells you about the dynamics of the cell gene therapy market. Multiple myeloma is a very severe illness involving plasma, which are part of the bone marrow, as you can see in the illustration, in some of our larger bones, as a form of cancer, and a very devastating form of cancer. Now, bear with me, this is a little complicated here. The two products that are have been approved, Obesame and Caricetti. I always have trouble with saying that one. Have a similar mechanism. You're going to hear the term anti-BCMA. That's B cell maturation antigen, which occurs on the multiple myeloma bad cells that are causing the cancer. So here's the process. This includes all the techniques we were talking about. Cells are harvested, blood products are harvested from a patient. The T cells, that's a form of an immunocell. The T cells are genetically modified by the CAR T process in a laboratory, and then they are infused back to the patient. These treated T cells are able to react with that antigen I mentioned, the BCMA antigen, and leads to the elimination of the multiple myeloma cells. Safety problems, well, some other cells may be expressing the same antigen. So a very complicated area, but nevertheless has been approved and appears to be helping a lot of severely ill individuals. But it's also a complicated regulatory path. There are regulatory discussions about when these expensive and complicated treatments should be provided to multiple myeloma patients. It should be when previous treatments failed or part of a separate regimen. So there's a lot of discussion within the literature and a lot of discussion about business implications because these are very expensive products. You will have to follow up with this. There's going to be an advisory committee in December of this year to discuss those particular issues. So in closing, I've given you a very strong overview of the process. There are some direct questions, Q&A on the slides that follow. Maybe we can take a quick look at one of them. Let's move to the next slide. This slide reinforces in a question of what we were talking about a little bit earlier, and knowing the understanding with the agency about autologous, where certain products are removed from one given patient and directly instilled back to that patient. So they have the separate regulatory pathway, and allogenic, where it's a donor, and it's important to understand how you would choose a donor. Next slide, please. And in this slide, how do we consider animal species? Well, you have to know the pharmacology of your proposed drug in a particular species. You have to know if that species would be susceptible to a viral vector, and what that viral vector does to the animal versus how it is expressed in the human being. There are occasions when there are no appropriate animal species, and those require significant discussions with the agency. So I think these are important things to consider. Another question will be on slide 21. If we could move there. The next slide, please. And as you can see, this again these come from some of the guidances, and this is about animal products. It plays into what we were saying about Nantucket, and there should be some compatibility uh, in what in cell therapy between the animal species and what might also be treated in humans. And the same for gene therapy, consideration of the characteristics of a vector. Please move to the next slide, please. The important question about control groups to think about. You're dealing with patients with severe illnesses and trying to demonstrate whether your product provides benefit and since patients are probably undergoing therapy, it would not be ethical to remove a baseline therapy from them. So in many cases, this would be an add-on therapy. And in other cases, you would use historical controls. And you can study some of the other options as you look over these slides later. Let's move on to the last slide, please. Well, one more slide there. Well, I've given you this overview. Most important considerations are the safety and development of a product that meets an unmet medical need, consideration of the off-target effects of cell and gene therapy, and understanding the regulatory pathway, and that there are more and more options available to you in approaching the agency and approaching them early, hopefully to have a more rapid review of your products, particularly if you have good data up front in your initial presentations. So again, Alan, Mindin, everybody in the audience, thank you for your help. Somewhere in there is my email if you have more questions, and hope we can all work together.